which are signals through which the body speak. As shown in table 1, we observe changes in blood parameters of rat treated with purified activity. Also, as shown in figure 4, we observe variation in body weights across experimental group, representing dose-dependent physiological response to the lectin, while figure 5 is a representation of the electrocardiograph showing a distinctive cardiac rhythm alteration, characterized by irregular RR interval and inconsistent spacing between successive waveform compared with the control group. These establish the influence of lectin on cardiac conduction and muscle physiology. Together, these findings establish that GALI contains a component that is biologically active with the ability to improve immune system and enhance health. Cloning ball leg 1. To translate discovery into application, Afolabi Balogun et al. 2012 used modern genetic techniques to isolate and characterize ball leg 1 gene to understand its architecture, genetic blueprints, and function. The result confirmed its membership in the mannose binding lectin family as shown in figure 6 and 7, a group with insecticidal property, and a close association with the snowdrop lectin as shown in figure 8 with a unique lobe motif that could underline its distinct activity as shown in figure 9. This was the first time garlic lectin gene was molecularly characterized in Nigeria, and a milestone that quietly placed my research within global conversation in plant biochemistry and remain widely cited till date. Vice Chancellor Ma, this taught me an enduring lesson that innovation is not the monopoly of location but conviction. Optimizing transformation. With our understanding of genetic composition of Bolek 1, Afolabi Balogun et al. 2011b, next focus was to assess its ability to confer protection in a different host, and our choice was an economic plant cotton. To do this, we tried out different laboratory conditions to enhance the DNA delivery into plant cells. Key observations include the realization that acetylsyringone addition, as shown in figure 10, improved agrobacterium virulence and DNA transfer into explant, Afolabi Balogun et al. 2014. Similarly, we observed that the explant age, as shown in figure 11, and the routine efficiency, as shown in figure 12, play a significant role in gene transformation, Afolabi Balogun et al. 2011b, 2013, and 2015. Vice Chancellor Ma'am, this was a taxing experiment. Each failed run was a classroom in itself, optimizing demand not only reagent but reflection and sometimes on one's own persistence. Through this phase, I learned that the beauty of molecular work lies not in the speed but in the precision, not in how many reactions one runs but in how deeply one understands the one that works. The implication of these findings, Afolabi Balogun et al. 2011, 2013, and 2014 affirm the benefits of garlic lectin extend beyond health. Specifically, Afolabi Balogun et al. 2011 he established the protective effect and cotton. My research naturally extended into other biological systems, reflecting a broad, multidisciplinary approach to using biotechnology for real-world impact. In indigenous chicken, Abdurrahim and Afolabi Balogun et al. 2016a. In tomato, Bello and Afolabi Balogun et al. 2016b. In tiger nut, Adeboyejo and Afolabi Balogun et al. 2017, and in guinea fowl, Shola Ojo and Afolabi Balogun et al. 2021. In essence, biotechnology bridged the laboratory and the land. Lessons from the formative year. Vice Chancellor Ma. These experiments taught me discipline, patience, and how small molecules can shape big questions. Working in the Department of Biochemistry at Ahmadu Bello University, Zaria, and later at the Agric prepared me for a broader challenge and ultimately guided my shift towards research with cancer diagnosis. The role of a mentor. Vice Chancellor Ma. You would agree with me that the path through academia is rarely linear, and like everyone, my trajectory include period of interruption and reflection that ultimately strengthen my focus and commitment to translational science. If early recalls represent the formative year of curiosity and training, the next phase of my journey mark a reawakening, the point where personal and opportunity converge, shaping this scientist I would become. Towards the conclusion of my doctoral studies, the demise of my husband, Afolabi Akim Olatunde Balogun, led to a temporary setback from active research. During this period, as Professor M.T. Bakari Odunola and the late Professor Andrew Jonathan Nock provided invaluable support, encouraging me to maintain engagement with science and envision future possibility. Their counsel and belief in my potential were pivotal in guiding my eventual return to science. This re-entry into active research was marked by participation in continental workshop, international collaboration that expanded both skill sets and scientific vision. Workshop in Etebe, Europe, and at the University of Ghana under the Creek Africa Network exposed me to advanced molecular biology technique, diagnosis, and bioinformatics, while connecting me to network of Africa and global peers. These experiences reinforce the importance of collaboration, resource sharing, and situating African science within global conversation. Technical and conceptual growth continue through short-time fellowship under the International Union for Biochemistry and Molecular Biology, IUBMB. Exchange visits with international peers while I acquire skills in multiplex immunofluorescence, confocal microscopy, tissue culture, and advanced bioinformatics. These capacities later became central to my research in oncology and infectious diseases, equipping me to address challenges relevant to low resource setting and public health priorities. Equally significant was a philosophical shift in my approach to science, where early curiosity-driven experiments from garlic purification to gene cloning focus on knowledge and knowledge generation. My new reform focus emphasized translational impact. My research began to focus not on scientific curiosity, but also by the imperative to develop solutions for community, improving maternal health diagnosis, enhancing molecular surveillance for antimicrobial research, and optimizing imaging solution adaptable to African context. This phase of my research crystallized three guiding principles: the value of mentorship, the strength of collaborative network, and the necessity to align research with societal need. These experiences collectively shaped my vision as a scientist, underpinning my approach as a clinical researcher committed to advancing both knowledge and practical solutions for Africa. It is against this backdrop that my research focused on leveraging biotechnology, which provide both the lens and tools to decode resistance mechanism and the design of rapid, reliable molecular diagnostics. Bench to benefits. In 2020, Bello and Afolabi Balogun et al. establish resistant gene among extended spectrum beta lactamase ESBL producing Escherichia coli isolated from pregnant women, implying the occurrence of bacteria carrying TEM-OX and other resistant gene in our antenatal patients. These are not just molecular names, they are signature of resistance. What does this imply? A pregnant woman carrying this resistant gene is unresponsive to antibiotic that the bacteria is resistant to. The implication is puts her at risk of persistent infection that could lead to kidney infection, sepsis, and preterm delivery. This publication provided the first molecular map of resistant gene in the population, underscoring the power of genetic surveillance in shaping antibiotic policy and preserving drug efficacy. Vice Chancellor Ma'am, for me, antimicrobial resistance research became more than the cataloging of resistant gene. It became a translational mission, equipping clinicians to prescribe wisely, informing infectious control policy, and protecting antibiotic effectiveness for future generation. Earlier efforts in antimicrobial resistance include molecular characterization of ESBL, Igwe and Afolabi Balogun et al. 2014, plasmid profiling of Staphylococcus aureus, Obajuluwan and Afolabi Balogun 2015, and molecular characterization of Panton Oji et al. 2016. And environmental biotechnology. Vice Chancellor Ma'am, leveraging on the theoretical knowledge that microbes are not completely agents of infection, but also powerful tool for protection and restoration, sets the foundation for my next research direction to solve environmental and public health challenges. Vice Chancellor Ma'am, this journey began at an unusual place, at the Osogbo Sacred Grove, simply because as a child I was fascinated by its reputation as an healing river. Can we remember the reading? Inuagural. And as a scientist, I later became fascinated by the ecology behind that belief. How microorganisms in natural ecosystem can purify water, transform pollutants, and maintain environmental health. In June 2016, Fountain University signed a memorandum of understanding with to analyze soil, water, and sediments of Osogbo Grove. Across our publication from 2020 to 2022, the data were consistent. The river contains trace elements, vitamin, detectable level of steroid hormone, as shown in figure 16 and 17, and its organism produce secondary metabolites in a nutrient-dependent pattern. Afolabi Balogun et al. 2021. Notably, the methionine zinc concentration was significantly elevated in sample collected from the ritual activity sites. Although this finding does not establish any therapeutic possibility, they provide a scientific lens for understanding how certain environmental characteristics may have contributed to the river's long-standing cultural perspective as beneficial. Ultimately, this work also allowed us to move beyond traditional and objectively characterized rivers chemistry and ecological health. Soil fungus isolates from the grove produce secondary metabolites, including nucleotide analog such as chlorogenic acid, tetrahydroquinone, and curcumic acid derivative with antioxidant, anti-inflammatory, and antimicrobial property as shown in figure 18. Additionally, the river's microbes is dominated by bacteria with strong potential for cleaning up pollutants as shown in figure 19, Afolabi Balogun et al. 2022. Taking together these findings offer the first molecular insight into a sacred ecosystem, framing the Osun Group not just as a cultural heritage site but as a biotechnological treasure. It is a place where rich biodiversity and significant discovery intersect, opening doors to environmental sustainability and the search for novel metabolites. Bridging traditional knowledge with molecular science. The discovery from Osun Grove broadened my perspective and pushed me to explore natural products more deeply as reservoir for therapeutic potential. Nigeria, like much of Africa, hold immense biodiversity and a rich heritage of indigenous medicine. Yet many of these resources remain overlooked simply because they lack molecular validation. Therefore, my work focused on filling this gap. To identify and characterize bioactive compounds from plants, fungi, and microbial system with relevance to mental health, oxidative stress, and ultimately cancer. Thus, laying the scientific foundation that later shaped my research in cancer and contribution with global impact. In collaboration with colleagues, we profile antioxidant and phytochemical properties of biogas slurry, Ajayi and Afolabi Balogun et al. 2001. Also at the agricultural residue, Olaniyan and Afolabi Balogun et al. 2025, demonstrating agricultural waste can yield pharmacologically active compound, turning environmental challenge into biomedical opportunity. Vice Chancellor Ma on validating folkloric uses of medicinal plants. Suleiman and Afolabi Balogun et al. 2024 turned to Mucuna puri, Yerekwe, a neuroactive African plant. Growing up, I knew how the seed of Yerekwe was used in preparing ero, an age-long remedy. Suleiman and Afolabi Balogun et al. 2024 established the plant extract of Mucuna puri produced clear improvements in Drosophila depressed model. It lowers oxidative stress level and supported the recovery of key neurotransmitters, including measurable antidepressant and neuroprotective effects, as shown in figure 20. This work not only established Mucuna purine as a promising therapeutic candidate, but also illustrates how traditional remedy, when examined through a molecular lens, can yield discovery of global biomedical relevance. The translational hack. Across these studies, a unifying message emerged. Through biotechnology, Africans' natural and cultural heritage gained a new voice in global science, transforming ancestral knowledge into evidence-based medicine and local discovery into universal benefits. Exploring the molecular signature of human papilloma virus. My transition from microbial and environmental biotechnology into molecular oncology was more than a change in discipline. It was a change in perspective. I moved from studying how microbes survive in environments to exploring how papilloma virus survive and adapts within us. Genome and erupt and disrupt as shown in figure 20, the natural mechanism that controls cell growth and development.